Hello viewers, welcome to this particular lecture on industrial microbiology where I shall be discussing some aspects of industrial fermentation. Here I shall be mainly covering the process of fermentation, the different products that is generated due to fermentation, the background of the process, the different fermenters used, and also the components used in fermentation process. I shall also discuss a practical example of citric acid production to understand the different nitty-gritties of the fermentation process. Now, fermentation actually is anaerobic respiration in which sugar is converted to alcohol or ethyl alcohol or organic acid in absence of oxygen under the action of microbial enzymes. And the process usually generates alcohol or organic acid, but at the same time, fermentation may produce different other products. And that include, it can be biomass generation, that is the microbial biomass. It can be production of different extracellular metabolites. It can be the production of intracellular metabolites like enzymes, and also it may involve transformation of the substrate, and this used substrate after fermentation may be used for other downstream processes. It is very important that this fermentation is carried out by microbes. Mostly it is bacteria, and at the same time it can be fungi or molds. Now, when it is bacteria, as we know, bacterial growth or the grand period of growth is divided into four major phases, namely the lag phase in which the organism is starting to grow. Then we come to the log phase where the organism is showing exponential growth. Then it comes to the stationary phase where it is not increasing nor decreasing, and then it goes into the reduction phase and into the death phase where the organism growth is totally gone and the organism is dead. Normally, during fermentation, we try to take the organisms in their log phase because the number of cells increase, and as the number of cells increase, the products generated will be more. Nobody actually knows when this process of fermentation started, because when we consider prehistoric times in the old Roman, Greek civilization, we have seen that they do have the beautiful technology of producing alcoholic beverage like beer and wine. And that means it is due to fermentation. As seen in this picture, that there are specific storage pots which is to store wine, produced due to bacterial or fungal fermentation. Of course, much later, it was Curie in 1917 that actually employed this fungus or this mold, Aspergillus niger, in the fermentation process for generating organic acid. And this is the fungus of Aspergillus with the conidia. Now, the most important factor is the culture media. The culture media where the microorganism will grow. The fermentation media should have the source of carbon, and normally this carbon source is brought about by different types of sugar like molasses, corn strip liquor, or it can be simple sucrose or glucose. And at the same time, it is having nitrogen source which is in the form of soybean meal, or it can be peptone. The source of potassium or the source of phosphate comes from potassium dihydrogen orthophosphate or potassium dipotassium monohydrogen phosphate. Depending upon the necessity, it is always important that we add some trace nutrients which include calcium, manganese, magnesium, iron, zinc. And if it is fungal media, then we need not have any bacterial growth, for which we are adding some antibiotics so that the bacterial contamination in the fungal culture is not there. After this particular microbes grow in the culture, we call it starter culture, it is scaled up and it is added to the fermenter. It is very important because this particular process will generate carbon dioxide, naturally there will be foam. So addition of antifoaming agent is very important in this particular aspect, where we are adding silica, and actually that silica gets into the air bubble and will rupture it. So basically antifoaming agent will increase the end product generated in fermentation. Now this is a typical fermenter. As you can see, it is a cylindrical structure where the temperature regulation is brought about by the jacket where there is water circulation. So whenever the temperature is generated, the temperature will come down. And at the same time there are specific inlet for culture, specific inlet for raw material, and outlet for the end product to be taken out, or the harvest pipe. If it is an aerobic particular fermenter, then there will be a sparger, and this sparger will circulate the air so that the air supply is uniform. But for anaerobic fermenter, the sparger is not required. So naturally, instead of sparger, we have an agitator. The purpose is same. Basically, it will agitate the particular solution, so the microbes will be always in motion and their activity increases, and thereby the fermentation product will also be generated. Now broadly, loading and generation, when we consider, it can be of two major types. One is the batch culture and the other one is the continuous culture. Actually, the basic difference is that this particular microorganisms are given in batches in case of batch culture, and the products which are generated are also taken from time to time. So naturally, this particular batch culture will require stages of input and stages of output. But on the contrary, continuous culture is a process where the nutrients are added and the cells are harvested at the continuous rate, where the volume of the suspension is kept constant and the production is more or less constant for a definite period of time. But of course, because the machine is continually in operation, it is very important to maintain close vigil, or we need to have trained personnel for the maintenance of the fermenter in continuous process. And as you can see, once the charge is given, the product will be generated continually. So that is an advantage also, where I am getting more product because of this continuous generation. Now it is very important that when any particular product is generated, it cannot be a single product. Like you can see, the cellulose which is pre-treated can actually be used for hydrogenation or can be used for fermentation to generate alcohol. Similarly, at a separate state, the whatever cellulose or the plant product that can be used for the generation of lignin, and after the fermentation, if the ethanol is generated, whatever is the used substrate that can be used in power generation through bioelectricity. So what we can see is, when we develop a particular biomass, this biomass at different steps will produce different end product. So this is definitely a very eventful process. It will not produce one type of product, it will keep on producing series of product, and also I am getting the end product at the end. So it is definitely very much profitable when we consider a particular product is generating large number of byproducts at the same shot or same go. Now, there are different products that are being generated by microbes in fermentation. First set of products, we call them primary metabolite. Why we call it? Because these products are more or less produced from the byproducts which are generated because of the metabolism of the cell. That means any common metabolic process like the respiratory process, like the Krebs cycle, like the EMP pathway will generate all these products. And these primary metabolites will include citric acid, which is produced by Aspergillus niger, glutamic acid, which is another amino acid, which is generated by Micrococcus, Corynebacterium, which is producing amino acids like lysine, threonine and tryptophan. Spirulina can be producing protein. Green algae can be producing pigments like beta carotene. So these are some primary products because these products are also being utilized by those particular microbes. But when we come to the secondary products, so you can see here, this is the common pathway where glucose is converted to acetyl coenzyme A, and this acetyl coenzyme A enters into the Krebs cycle, and the byproducts of the Krebs cycle or the EMP pathway will generate series of these amino acids like lysine, alanine, tryptophan, asparagine, isoleucine, proline, etc. So thereby we do not really have to have some special input to get all these products. But when it comes to secondary metabolite are the special product. Of course, the center is the acetyl coenzyme A, which can produce toxins like ochratoxin, which can produce amino acids, and from amino acids we can get specialized product which are antibiotics. We can produce mevalonic acid, and from mevalonic acid we can produce hormones like gibberellic acid and abscisic acid. So at the same time it can produce different other antibiotics or different other antimicrobial product which are not normally produced by the organism when grown in a suitable constant medium. Now we come to the specific example of citric acid. Why I am considering it? Because if we consider a specific example, then we can understand that what actually fermentation is or how it is carried out. So citric acid is a very common product which is produced by the citric acid cycle, which is a primary metabolite, C6H8O7. And this particular metabolite is produced by all living organisms. But what is important, that this is produced by microbes which are not always utilized. So naturally this can be harvested for animal use or human use. And technically 60% of citric acid may be used in the food industry, and the rest 10% can be pharmaceuticals, and the other 30% can be in other different types of industries. Citric acid commonly is produced by or from glucose, which is converted to acetyl coenzyme A, and this acetyl coenzyme A will enter the Krebs cycle producing citric acid, and the end product is oxaloacetic acid. Or sometimes glucose is directly converted to pyruvic acid. Pyruvic acid forms the oxaloacetic acid, which ultimately is converted to citric acid. Now really it is a very important matter that all these metabolites are not always produced by the organism. So some of these products, some of these organic acids, some of these vitamins are produced by chemical process. Now we have to consider that whether it is the chemical process or whether it is the fermentation process which is more beneficial for me. If the product is generated by chemical process and the cost incurred is more, then I am naturally not going into the fermentation process because one-time input is more. But if I am seeing that the chemical process is too costly, then I get into fermentation product. And specifically this particular citric acid production for a great number of years was produced chemically. But now we have understood that okay, the yeast and molds can produce citric acid by fermentation. Similarly, there may be some vitamins like vitamin C, vitamin B2, which are produced both by chemical as well as by fermentative process. So citric acid production by industrial process was denoted by Carl Shields in 1874 in England. And actually the raw material used was lemon juice. But subsequently Weber in 1923 could explain that actually it is Penicillium glaucum, which is a mold fungus, can produce citric acid, and this is produced as a byproduct in the way of producing calcium oxalate or oxalic acid. And naturally from that time onwards we could understand there may be different types of microbes involved in citric acid production, and that include the fungi like Aspergillus niger and other species of Aspergillus, yeast like Saccharomycopsis lipolytica, Candida tropicalis, bacteria like Bacillus licheniformis or even Bacillus subtilis. So there may be host of different organisms that is employed in citric acid production, reported by different authors at different times. But broadly, the raw material that is being used for citric acid production can be classified in two distinct types, which are low ash content, which include the cane or beet sugar, or even dextrose syrup, and also the other one which high ash content, which include the beet molasses. And this is the key organism, that is the Aspergillus niger, which is the common mold that can produce this citric acid by fermentation. Now there may be different types of fermentation process, which is also very important to understand. This can be liquid fermentation, it can be solid state fermentation. Liquid fermentation can also be submerged fermentation or surface fermentation. Now in case of submerged fermentation or SMF, actually the organism is growing inside the medium. So naturally it has to have a stirred fermenter where the organism is continually stirred so that the utilization of the medium is better. But surface fermentation on the other hand is actually not being employed by anaerobic organisms. It can be partially aerobic ones so that it grows on the surface. And in this particular case, they are using specialized tray or specialized container with a large surface area. So organisms can grow in multiple and utilize air and at the same time can produce the fermented product below. This is the typical container where the surface fermentation is being carried out to produce citric acid. But when we come to the solid state fermentation, it is an alternative method used in agro-industrial process, and commonly this process was developed in Japan, not for citric acid though, but it is called the common koji process to generate soya sauce. And here the organism is actually Aspergillus. Now what is happening, that the particular whatever, it can be rice, it can be barley, which is starch enriched product, which is commonly being inoculated with a particular organism, followed by fermentation, then distillation and aging and dilution and bottling. Now why I am telling this? This particular process of koji became very important because it is a food additive. Now we put the same process in fermentation of citric acid, where there will be the substrate. In the substrate, the inoculum of Aspergillus will be given. The fermentation product will be there, followed by purification and dilution or bottling. Rather, it is a very simple process in food industry. And it is very important, as I have considered for general fermentation, citric acid medium or the organism growing in the medium for producing citric acid should have carbon in the form of sucrose and glucose, nitrogen in the form of different ammonium salts, phosphorus in the form of potassium dihydrogen orthophosphate, and certain trace elements like zinc, iron, manganese, magnesium and copper. These are very important constituents to be added to produce citric acid. Like any other process, there will be certain inhibitors, because sometimes if the process is too fast, I can add some fluorides, which can be sodium or calcium, and this process will reduce the process. So sometimes inhibitors are equally important to reduce the process. And there are certain other factors which has to be considered, which include the pH, which is of course in the acidic range around 4 to 5, which includes the supply of alcohol. If the alcohol content is high, naturally citric acid production will go down. So we have to select a particular strain which will produce low alcohol. And finally, and most importantly, is the recovery of the product, where I am producing series of compounds, but I need to recover citric acid. It will include precipitation, extraction, adsorption or absorption, so that I am getting the maximum citric acid from the mother liquor, and I can actually purify it from other bacteria by passing it through activated charcoal or passing it through cation or anion exchanger. And that is the most important process by which I will generate the finished product which will be fit for storage. So viewers, in this particular lecture, I have tried to discuss the major aspects of industrial fermentation, what are the steps involved in this fermentation process, what are the different products that we can generate, what may be the difficulties, and also I have taken a concrete example of citric acid and I have tried to explain the process of industrial fermentation. Thank you.